So I'm not sure if we are live now.
Yes, I think.
Okay. All right. So they tell us that we are live. So good afternoon, everyone. Welcome to this session. So for the next 1.5 hours, we are going to have presentations about bioenergetics, metabolism, metabolomics of pancreatitis and pancreatic cancer. So kind of looking at the big picture. My name is Federico Canzian from the German Cancer Research Center. I will be co-chairing this session together with Josef Mallet and Alexei Tipikin. And we should get started with our first speaker, who is Jason Bruce, who is going to talk about fueling calcium pumps in pancreatitis and pancreatic cancer. So, Jason, please.
Good afternoon. First of all, I'd like to thank the organisers for the kind invitation and also for organising a fantastic conference under the very difficult circumstances. So today I'm going to be talking about cellular metabolism and the regulation of cytosolic calcium. Central to this is the plasma membrane calcium pump, which is an ATP-driven pump that regulates normal cytosolic calcium. Cutting off this ATP supply to the PMCA essentially leads to a cytotoxic calcium overload and cell death, essentially in all cells. But what I hope to convince you is that in pancreatic cancer cells, glycolytic ATP supply to the PMCA is critical for cell survival. And cutting off this supply with glycolytic inhibitors leads to this toxic calcium overload and cell death. Also, during pancreatitis, endogenous insulin secretion protects against pancreatic injury. And again, this leads to an increase in glycolytic flux, which protects against pancreatitis. So I don't really need to convince this audience that the survival statistics for pancreatic cancer are very grim indeed. And this is because this disease is asymptomatic, but it's also because the tumor is highly desmoplastic, which makes delivery of cytotoxic drugs very difficult. So there are three key strands of research that are likely to improve treatment. First of all, early detection with improved biomarkers. Secondly, improved drug delivery by targeting the extracellular matrix. But I would also argue that the development of novel cytotoxic drugs that harness various vulnerabilities of pancreatic cancer cells is likely to be an important research strategy moving forward. Now, as many of you will know, all cancer cells exhibit an altered metabolism due to a decrease in mitochondrial metabolism and an upregulation of glycolysis. Now, this might be an oversimplification because we know that mitochondria remain active, but what we do know is that the upregulation of glycolysis provides a number of survival advantages for the cancer cells, most notably the accumulation of biosynthetic glycolytic intermediates important for cell growth. Extracellular acidification, which occurs due to lactic acid efflux, also facilitates matrix metalloproteinases, important for migration and invasion, but also suppresses cancer immunity. However, ATP production and the maintenance of the calcium pump is also critical for cell survival, and inhibiting glycolysis leads to this toxic calcium overload and cell death. We can measure PMCA activity by depleting the the ER calcium stores and activating store-operated calcium entry. And this leads to a huge increase in cytosolic calcium, and the PMCA now is working at full tilt. We can remove the external calcium and monitor the calcium clearance rate, which is due to PMCA. And we can apply a second clearance phase and apply various treatments to assess the effect on PMCA activity. We can also assess the metabolism in numerous ways, most notably the Seahorse XF analyzer, which allows us to measure oxygen consumption rate as a measure of mitochondria, and extracellular acidification rate as a measure of glycolysis. We can also measure ATP within the cytosol using GO18, which is a FRET-based fluorescent reporter, which changes color as it binds ATP. And now, if we treat the cells with oligomycin, which inhibits the mitochondrial ATP synthase, no, not surprisingly, there's a reduction in oxygen consumption rate and a corresponding increase in ECAR, which preserves cellular ATP. However, if we now inhibit glycolysis with iodoacetate, we see a rapid drop in ECAR, but this compensatory increase in OCAR is insufficient to preserve cellular ATP, and this is depleted. Now, if we look at what happens to PMCA activity, we see that oligomycin has no effect on PMCA, whereas iodoacetate inhibits the PMCA. This leads to cytotoxic calcium overload, whereas oligomycin has no effect. And similarly, using different inhibitors, inhibition of glycolysis with bromopyruvate causes necrosis, whereas inhibiting mitochondria with CCCP has no effect. So we next wanted to determine the key glycolytic enzymes, and pyruvate kinase M2 is overexpressed in pancreatic cancer and leads to poor patient survival, and this is responsible for providing ATP. PFKFB3 is another key enzyme which produces fructose 2,6-bisphosphate, which is an allosteric activator of PFK1, which is the rate-limiting step of glycolysis. And both of these enzymes are also overexpressed in pancreatic cancer and lead to poor patient survival. We can inhibit both of these enzymes. First of all, we can inhibit PKM2 using chiconin, and this is derived from Chinese herbal remedies, but it has a high selectivity for PKM2. This reduces cell growth. It also reduces cell viability. It induces cell death as assessed by part 1 cleavage, and it also inhibits cell migration. We can, the mechanism for these effects, not surprisingly, are due to an effect on glycolysis. We also see ATP depletion and inhibition of the PMCA and cytotoxic calcium overload. We can also inhibit PFKFB3 using the drug PFK15. And this also inhibits cell growth, reduces cell viability, induces cell death, inhibits PMCA activity, and causes cytotoxic calcium overload. But you'll notice that this drug has no effect on human pancreatic stellate cells, both in terms of calcium and cell death. And the reason for this is that this enzyme is not expressed in these cells, suggesting that this drug is specific for this enzyme. We also noticed that many of these glycolytic enzymes are associated with the plasma membrane, where they might be providing a privileged ATP supply to the PMCA in close proximity. There's also evidence that PKM2 can exist as a high activity tetramer producing lots of ATP and pyruvate, or a low activity dimer, which causes a bottleneck at the terminal end of glycolysis and accumulation of biosynthetic intermediates. There's also evidence that PKM2 can shuttle from both high activity tetramer to produce bioenergetics, or low activity dimer to meet the biosynthetic demands of the cell. And this can occur at different stages of the cell cycle. So during G2M, there is tetrameric PKM2, which is important to provide a boost of cellular energy for cell division, whereas the low activity dimeric PKM2 is important during G1S for biosynthesis. This means that if we arrest the cells during G2, we can effectively shift the cells towards G2, and this causes a potentiation of both ATP depletion and cell death. Now, this suggests that the combination of a CDK1 inhibitor and a PKM2 inhibitor might be an effective way of selectively killing pancreatic cancer cells. I'd now like to switch to pancreatitis, and I think many of you will know that pancreatitis is caused by excessive alcohol and fat consumption and bile acid reflux from gallstones. Both bile acids and fatty acid ethanol metabolites impair mitochondrial function, leading to ATP depletion, calcium overload, and necrotic cell death. And what we've demonstrated over the years is that insulin has a protective effect on virtually all of these steps. There's a very strong link between diabetes and pancreatitis, not least because pancreatitis causes injury, collateral injury of the pancreatic beta cells, which leads to a loss of insulin secretion, and this is known as type 3C diabetes. There's also evidence that pre-existing diabetes can make pancreatitis worse, and the mechanism for this was unclear, as it could be due to impaired insulin secretion or it could be due to hyperglycemia. And this was unclear until we discovered that insulin actually has a direct protective effect on asthma cells due to preservation of cellular ATP and maintenance of low cytosolic calcium. Therefore, during diabetes, when there's a loss of insulin secretion, this protective mechanism is abolished, leading to a much more severe acinar cell injury and presumably escalation to a more severe acute pancreatitis. Now, we demonstrated this using cellular models of pancreatitis in which we treated acinar cells with palmitoleic acid, a pancreatitis-inducing agent, which we found insulin prevented ATP depletion, inhibition of the PMCA, calcium overload, and necrotic cell death. And we also recently discovered that these effects were abolished when we isolated acinar cells from Pasurco mice in which the insulin receptor has been specifically deleted from pancreatic acinar cells. The mechanism involves a switch from mitochondria towards glycolytic metabolism, and we assess this by measuring NADH autofluorescence. So the bulk of NADH comes from the Krebs cycle within the mitochondria. So when we deplete this with a metabolic uncoupler, we get a drop in NADH autofluorescence. But also there's a source coming from glycolytic enzyme GAPDH. And therefore, if we inhibit GAPDH, we get a further decrease in NADH. And interestingly, when we treat with insulin, there's a reduction in mitochondrial NADH and an increase in glycolytic NADH. And this is further supported if we measure magnesium green. This is an indirect measure of ATP because most ATP within the cell exists as magnesium ATP. So when ATP is depleted, you get an increase in magnesium fluorescence, and therefore, using the same experimental strategy, we can deplete mitochondrial ATP with CCCP and then deplete glycolytic ATP with iodoacetate. And again, similar to NADH, insulin reduces mitochondrial ATP depletion and increases glycolytic ATP. The molecular mechanism for this involves AKT-mediated phosphorylation of the key glycolytic enzyme PFK-FB2, which increases fructose 2,6-bisphosphate, thereby driving glycolytic metabolism and the preservation of cellular ATP, important for regulating PMCA function. So this is very similar to what we see in pancreatic cancer cells, except this specific isoform, PFK-FB2, is a tunable enzyme and is increased when phosphorylated by AKT. So in other words, this acts, if you like, as a volume control in glycolysis. And as you can see, not only does insulin increase AKT phosphorylation, but it also increases PFK-FB2 phosphorylation. We can also exogenously administer insulin using the hyperinsulinemic euglycemic clamp and combine this with cerulean infusion-induced pancreatitis. So we can elevate insulin to quite high levels, around about 3 to 4 times higher than what the animals ever see. But we can also tightly control the glucose concentration by detecting glucose every 15 minutes and then altering the glucose infusion rate. If we then combine this with cerulean infusion, we can detect plasma amylase levels very regularly over this 5-hour period. And as you can see, as you would expect, at 5 hours, cerulean increases plasma amylase. But the take-home message is that if we apply the insulin infusion or insulin clamp 2 hours into this, then we can see a significant reduction in plasma amylase, suggesting for the first time that insulin might have a therapeutic utility for the treatment of pancreatitis. But admittedly, this is only in healthy mice. Whether this will translate to critically ill patients with severe acute pancreatitis that are already metabolically compromised is somewhat contentious and debatable. However, what it does do is open up the possibility of developing insulin mimetic drugs that will mimic some of the downstream signaling pathways and mechanisms that I've shown previously. So to summarize then, insulin protection during pancreatitis is caused by AKT-mediated phosphorylation of PFK-FB2, which drives glycolytic flux, maintains cellular ATP and PMCA function, even in the face of impaired mitochondrial function. During diabetes, this protection is effectively abolished, leading to both ATP depletion, calcium overload, and necrotic cell death, and presumably a much more severe acute pancreatitis. And what it does is opens up the possibility that insulin mimetics that mimic this molecular mechanism might be a potential therapy moving forward for acute pancreatitis. So this leaves me to thank the people that did the work, both past and present members of the lab, both on the Pancreatic Cancer Project and on the Pancreatitis Project. I would particularly like to thank John Williams from the University of Michigan and also the Medical Research Council. Thank you very much for your attention, and I'd be happy to take any questions.
Right, thank you, Jason, for this comprehensive presentation. Very interesting.
Thank you.
And so now we can have some questions. I see one from the audience in the chat. So Prince Alawadi is asking, do you think Bruton kinase inhibitor can reduce the calcium influx in pancreatitis by downregulating downstream signaling pathways related to calcium?
Okay, thanks for the question. I'm not so sure I fully understand the question. Is that supposed to be protein kinases, or is there a specific Bruton kinase? I don't know what that refers to, but I think protein kinases in general are known to regulate various calcium transport pathways. I'm not aware of any specific kinase regulation of ORI1, which is perhaps the most important channel in pancreatitis. Any effect of any kinase inhibitor, I would predict, would be rather subtle at regulating the cytotoxic calcium overload that facilitates the damage associated with pancreatitis. And perhaps the best approach would be to simply inhibit that channel, which has been shown to be very effective as a treatment of pancreatitis, at least in animal models and in cellular models, as shown by Robert Sutton's group and Ole Peterson's group, among others. So, so going back to the question, I'm not sure exactly what the question was asking, but I think in terms of protein kinases, I think any inhibitors are likely to be rather subtle.
Okay, thank you. Then we have another question by Peter Hegi.
Yeah, hi Jason,
Hi.
brilliant presentation as usual, congratulations.
Thank you.
You most probably will be not surprised my question concerning the pancreatitis and diabetes part. Actually, we can see in the world plenty of times that there is really big hyperglycemia for the patients, and hyperglycemia is really very toxic for the cell size. So hyperglycemia is associated with really severe pancreatitis. So currently we try to reduce it by insulin, external insulin to give. But
Okay.
do you think it could be maybe in the future other method to give, for example, directly to the pancreas somehow, deliver insulin or prokinins? So what do you think concerning the external insulin? That if, for example, for the patients with suffering pancreatitis, you give insulin plus glucose as well, would it help?
Well, as I showed in the animal models, infusing insulin using the hyperinsulinemic euglycemic clamp, which allows a very high level of insulin infusion whilst maintaining constant glucose control, was very effective, at least at reducing the early stages of injury as assessed by plasma amylase. Translating that to severe pancreatitis patients, I would predict, and I don't know, I've not seen any of your data yet from your studies, but I would have predicted that that might be a little bit more challenging because severe pancreatitis patients are severely metabolically compromised. They are likely to be at high risk of ketoacidosis. So maintaining euglycemia is likely to be very challenging. And so I don't know the answer, to be fair, and I think localized perfusion might be important. And the reason for that is that the asthma cells require about 10 times higher insulin than the systemic circulation. So that's adds to the challenge. But I would argue that perhaps developing insulin mimetics, and if you look at the particular molecular mechanism that we've identified, the phosphorylation of PFK-FB2, the glycolytic enzyme, that same phosphorylation site is shared by AMP-dependent kinase. And AMP-dependent kinases are activated by metformin, which is an anti-diabetic drug. So I would argue that perhaps this is the best approach, is to try and mimic the downstream signaling, mimicking the phosphorylation of PFK-FB2, either with pKa or with AMP kinases. And of course, I think Robert Sutton several years ago, along with Ole Peterson, showed that caffeine, which obviously is an inhibitor of phosphodiesterase and therefore will elevate pKa, has a protective effect. And so that could be the reason why, because it's pKa phosphorylation of PFK-FB2 that might be having that protective effect. So I would argue that that might be the best approach, and this is something that we're currently working on.
Thank you very much, Jason.
Thank you.
Thank you. So we are still on time, right on time, so we should move on. Also, I don't see any other questions, so I pass the microphone to my colleagues who will introduce the next speaker.
Okay, thank you very much. So it is my pleasure to introduce Professor Ioanna, who is one of the field leaders in studies of cellular signaling in pancreatic cancer. And with this, Professor Ioanna, the floor is yours.
Thank you very much to the organizer for giving me the opportunity to present our work in this singular APC 2022. The title of my talk is The Cellular Stress Response as an Efficient Therapeutic Target for Treating Pancreatic Cancer. 25 years ago, we started a systematic approach with the aim to identify the genes activated in the pancreas in response to the stress. To do this, we induced an experimental acute pancreatitis in the mice and extracted the pancreatic RNAs, which were transformed in cDNA and cloned into plasmids. We sequenced more than 2,000 clones, from which we analyzed their sequence and focused the interest on the novel and original genes. We systematically studied the expression of all these new genes and selected NUPER1, which show a rapid, strong, and transient activation during pancreatitis-induced cellular stress. My talk will focus on the results we have obtained with this gene as a target to treat pancreatic cancer. First of all, I will take a few minutes to explain why your project is original. It is original because it takes advantage of the stress responses induced in cancer tissue. How the stress is activated? On one hand, as a consequence of the oncogenic transformation, which is known as oncogenic stress, and on the other hand, as the response to the stress induced by the progressive decrease in nutrients and oxygen through the abnormal blood vessels, which is known as metabolic stress. Therefore, a very strong stress is induced in cancer cells, which help the cells to survive. The rationale of our strategy is based on the fact that the stress induces a response which allows the adaptation of the cancer cell, and consequently, the cell survives, and therefore the tumor can progress. If we are able to inhibit this stress response, we should inhibit the cellular adaptation, and therefore we can stop the tumor progression. Here you can see on the right part of the expression of NUPER1 protein is strongly activated in cancer cell, but its expression is undetectable in the healthy tissue on the left part of the slide. This is why this protein could be a good candidate to be used as a target to inhibit tumor development. As a proof of concept, in 2011, we inactivated NUPER1 as a pancreatic cancer cell by using a genetic approach and demonstrated that tumor development was completely blocked in xenograft model. Then several laboratories around the world applied our strategy and demonstrated comparable results on several other cancer models. Therefore, NUPER1 seemed to be a good target for treating pancreatic cancer, but also probably other cancers. The problem is that genetic inactivation is not yet applicable efficiently in patients presently. Then we tried to identify small compounds inhibiting NUPER1. NUPER1 is a small protein of 82 amino acids, and structurally is an intrinsically disordered protein, or IDP. Therefore, conventional drug development methods cannot be applied in this case. Consequently, as a first step, we developed a sophisticated screening method based on the fluorescence thermal denaturation and found that 3-fluoperazine, a drug which has been used to treat schizophrenia for several decades, bind and inhibits NUPER1. The second step was to determine the structure of the complex NUPER1 with 3-fluoperazine, and we found that this interaction involves amino acid in position alanine 33 and threonine 68. The third step consisted to optimize the structure through the combination of multiple in silico, in vitro, and in vivo approach. Using this systematic approach, the strategy, we developed a new compound which was named ZZW115. We have evaluated the anti-cancer activity of the ZZW115 on 10 primary cells derived from PDAC and a commercially available cell line, MIAPACA2. By treating this cell with increasing concentration of ZZW115, and observed that the EC50 is around to 1 to 3 micromolar. Then we evaluated its anti-tumor effect on several xenograft models of pancreatic cancer and found a dose-dependent effect with a maximal effect at 5 milligrams kilo per day. And which is of clinical relevance is that we didn't observe any obvious toxic effect. Remarkably, cancer cells of no pancreatic origin, such as those derived from melanoma, colorectal cancer, liver cancer, lung cancer, bladder cancer, osteosarcoma, leukemia, and lymphoma, breast cancer, and glioblastoma, are also sensitive to the in vitro to the treatment with ZZW115 and in the same range of sensitivity that of the pancreas cancer cell. The take-home message is NUPER1 is an excellent candidate to be targeted by small compounds for treatment, unless pancreatic cancer, but also probably other cancer. But how ZZW115 acts as an anti-cancer agent? What is its mechanism of action? We started the study of the mechanism of action trying to identify the partners of NUPER1. For that, we transfected a pancreatic cancer cell line with a vector expressing the flagged NUPER1 protein. Then we performed a co-immunoprecipitation with an antibody directed against the flag, and the product was analyzed by liquid chromatography combined with tandem mass spectrometry, followed by a bioinformatic analysis. Surprisingly, we found that 69 proteins were identified as in nuclear transport, and 263 were proteins with nuclear function. Among the proteins that would interact with NUPER1, we find importins, which are a transporter of the nuclear proteins from the cytoplasm to the nucleus. An analysis by PLEA, proximity ligation assay, which is used to study the interaction between two proteins, showed that NUPER1 binds to importins as demonstrated in the fuchsia dots in this slide. Interestingly, the treatment with ZZW115 inhibits completely this interaction. NUPER1 has an NLS sequence, NLS for nuclear localization signal in its C-terminal region. Importins bind to NUPER1 through this sequence, lets its nuclear translocation and permitting the tumor development. Our structural study showed that ZZW115 binds to NUPER1 through amino acid alanine 33 and threonine 68. And the threonine 68 is located into the NLS sequence. The affinity of the ZZW115 is greater than from importin, so its binding to the NLS displaces the binding for importins, preventing its cancer translocation and consequently the tumor development. Immunofluorescence analysis revealed that NUPER1 is of nuclear localization as expected, but the treatment with ZZW115 blocks completely its translocation. In presence of ZZW115, NUPER1 remains outside of the nucleus. The next question was how the inactivation of NUPER1 kills the cell. Inactivation of NUPER1 by knocking down or by using its pharmacological inhibitor ZZW115 induces a mitochondrial catastrophe. You can observe at the bottom left the ultrastructure of the normal mitochondria and the mitochondria after the treatment with ZZW115. As a consequence of the catastrophe, we observe a concomitant decrease in production of ATP, the respiration of the mitochondria, and a strong production of ROS. As a consequence of these effects, cancer cells show strong activation of caspase and cell death by apoptosis, which is partially inhibited by the pan-caspase inhibitor ZBAD FMK. We also found a strong release in LDH, a marker of necrosis, which is partially inhibited by necrostatin and suggesting a cell death by necroptosis. And finally, the accumulation of ROS, which induces an hydroperoxidation or lipids, induces cell death by ferroptosis, which is partially inhibited by the ferrostatin. The co-immunoprecipitation assay also demonstrated that PARP1 is a partner of NUPER1. The interaction was validated by the PLA assay, as you can see from the green dots in this slide. And most importantly, this interaction is completely sensitive to the ZZW115 treatment. How can we associate the mitochondrial catastrophe with the PARP1 interaction? PARP1 is an enzyme activated by several cellular stress whose function is to pyrylate proteins. Pyrylation is a post-translational process which, when uncontrolled, kills the cell. Studies carried out in vitro in a cellular system demonstrated a dose-dependent effect of NUPER1 as an inhibitor of PARP1 activity. Our working hypothesis was that NUPER1 is a negative regulator of PARP1 activity and that ZZW115 prevents this function, which results in a global hyperpyrylation with its terrible effect on cells. If so, olaparib, a specific pharmacological inhibitor of PARP1, should inhibit the effect of ZZW115. Mitochondrial hyperpyrylation could be responsible of the ZZW115-induced mitochondrial catastrophe. Treatment of ZZW115 induces a very clear disorganization of the mitochondrial network, visualized with the mitotracker stained in red here. The disorganization is prevented with the olaparib, as presented here. Oxidative mitochondrial respiration was assessed by the seahorse, as presented at the top and right of the slide. ZZW115 strongly inhibits respiration, which is reversible in a dose-dependent manner by olaparib, which is represented in green. Finally, the production of ATP is significantly reduced by the treatment with ZZW115, but it remains normal if the cells are simultaneously treated with olaparib. We can conclude that the mitochondrial catastrophe observed after treatment with ZZW is at least in part due to the hyperpyrylation of the mitochondria. To validate your hypothesis, you can see the treatment with ZZW115 induces this hyperpyrylation as revealed by the immunofluorescence in cells treated with ZZW, but is completely rescued by olaparib treatment. We can conclude that NUPER1 binds and inhibits PARP1 activity in vitro in a dose-dependent manner, NUPER1 inactivation through the ZZW115 treatment induces hyperparylation. The mitochondrial network is strongly altered in cells treated with ZZW115, which is completely rescued by olaparib. The mitochondrial oxidative respiration is reduced in cells treated with the ZZW115 and rescued by olaparib in a dose-dependent manner. Deficiency of ATP production as consequence of the ZZW115 treatment is rescued also by olaparib. ZZW115 treatment induced hyperparylation, which is rescued by olaparib. And finally, the mitochondrial catastrophe as consequence of the hyperparylation induced cell death by apoptosis, necroptosis, and ferroptosis concomitantly. As a final conclusion, we can say that targeting the stress protein NUPER1 with the pharmacological compound ZZW115 is a promising strategy for cancer treatment, and its mechanism of action for killing the cancer cell is really original. I would like to thank to my collaborators over the last years and the founder of our research. Thank you very much for your attention.
Okay, thank you very much for this excellent presentation. I think that one of the questions was already answered during the talk. I actually asked it before you discussed the mechanism. And there is another question in the chat. It is from Professor Krivoruchka. And it is a longish question, but I maybe rephrase it briefly, that he postulates that there is increase of generation, presumably reactive oxygen species, could be increase of generation of reactive oxygen species, which will cause cellular damage, and whether this will affect your potential treatment.
Yes, thank you very much for this question. It is an evident question. But the advantage of this system is that NUPER1, which is targeted by the ZZW115, is specifically produced in the transformed cell, not in normal cell. When we treat normal cell with the compound, we have almost no effect. This is specifically expressed in is a mechanism which is developed during the transformation of the pancreatic cancer cell, but probably other cancer cell, which remain specific. This is why we don't observe when we treat animals systematically with the ZZW, we have no effects in all the organs. We found that completely reduction in the mass of the tumor. Thank you for the question. This is important.
Okay, excellent. So I think that there is just maybe general question about the this mitochondrial catastrophe.
Yeah.
So what you show is, of course, a drastic rearrangement of the mitochondria, drastic changes of the, but presumably loss of mitochondrial membrane potential should happen actually even earlier.
Yeah, okay, this is also a evident question because it's not clear. But in our hands, when we treat the cell with the pharmacological compound to inhibit NUPER1, we found that hyperparylation, and hyperparylation is good for DNA repair, but it's bad for the cells, because when you hyperparylate normal proteins, you can inactivate some of them, which is a systemic effect on all the proteins, particularly concentrated around the membrane of the mitochondria, and mitochondria became inactive. This is why when we treat the cell with olaparib, which is a strong inhibitor of the parylation of the cell, we found that we can recover this effect. I know, I'm not sure that this is clear, my response.
No, it is clear. It is just this is the concept which I'm not familiar. So, but yeah, very interesting. Thank you. Okay, I think that
It's time to it's time to move forward with
the next.
yeah.
It's time
to move forward with the program. It is my great pleasure to introduce Edne Costello, professor of molecular oncology from University of Liverpool, and she will talk about using new onset diabetes for early detection of pancreatic cancer. So please, the stage is yours.
I would like to thank the organizers for the opportunity to present on the topic of using new onset diabetes for early detection of pancreatic cancer. I'm Edne Costello. I'm a research scientist and I work at the University of Liverpool. This is my financial disclosure. Pancreatic cancer, as I think everybody attending this meeting knows, is a deadly disease and the incident rates are increasing. The overall 5-year survival is approximately 9 to 10%. What is really clear is that for individuals who have whose cancer is operable, the survival is much better. The 5-year survival for patients with operable pancreatic cancer is approximately 32%. If the disease is spread locally, the 5-year survival drops to 12%, and it drops significantly for individuals for whom the disease has spread to distant organs. So for patients and families where in 10% of cases there's a family history of pancreatic cancer and screening programs are available. There is, however, no screen or test that allows earlier detection for the remaining 90% of cases. I'm talking about sporadic pancreatic cancer. So what are the barriers to asymptomatic screening of the general adult population for pancreatic cancer? The most significant barrier is low incidence of disease. There are approximately 13 pancreatic cases per 100,000 individuals. If you go into the group of over 50-year-olds, that increases to 37 cases per 100,000. So even highly specific tests in this group could lead to far more false positives than true positive screening results. And false positive test results cause worry for the individual who's been given that result. They require subsequent diagnostic evaluation that is unnecessary and potentially harmful. And of course, they come with considerable cost to the healthcare system. So while screening in the asymptomatic adult population is not possible at the moment, screening in high-risk individuals is recommended. And the high-risk group that I would like to focus on today is new onset diabetes. So let's look at the relationship between diabetes and pancreatic cancer. Individuals with long-standing diabetes, that is diabetes of more than 5 years duration, carries an increased risk of pancreatic cancer of 1 to 1.5 fold. Individuals with diabetes of less than 1 year duration, for those individuals, the relative risk of pancreatic cancer increases to 6 to 8 fold. And the probability of developing pancreatic cancer in 3 years is 1%. So this is the high-risk group that is the focus of my presentation. Hyperglycemia begins prior to pancreatic cancer being visible on imaging and is associated with small tumors, can be associated with small tumors. So let's have a look at the picture that has emerged. And this picture is modified from work done by Shari et al. So as I said, hyperglycemia beginning 36 months prior to cancer diagnosis, shown here with the red line. At about 6 to 12 months prior to cancer diagnosis, hyperglycemia tips into diabetes. So if it was possible at the time of diagnosis of diabetes to diagnose pancreatic cancer, this would enable early detection or early diagnosis of pancreatic cancer. It would bring the diagnosis of cancer forward. And it would potentially have positive effects for a large number of pancreatic cancer patients. More than 50% of pancreatic cancer patients have diabetes, 30% have new onset diabetes, making it the highest risk group for pancreatic cancer. So I'm happy to say that there are a number of studies now aimed at understanding new onset diabetes for pancreatic cancer detection. And these are mostly cohort studies. There are 2 studies in the United States. There's a study in Hungary and our own study in the United Kingdom, and I'll refer and present a little bit more about that as I go along. So these are cohort studies designed at generating resources and in terms of biobanks of samples, and also information and clinical data on individuals, all aimed at understanding better the probability of pancreatic cancer in this cohort and how it might be possible to intervene and detect the disease earlier. So new-onset diabetes is common, with more than 200,000 new cases every year in the UK, for example. And the incidence of pancreatic cancer in this group is 1%. So 1% of individuals newly diagnosed with new-onset diabetes has pancreatic cancer. So it's not feasible with current modalities to screen all individuals with new-onset diabetes for pancreatic cancer. So if we look more closely at this group, we see that 10% of individuals in this group actually have diabetes secondary to pancreatic disease or type 3C diabetes. So if there was a way to enrich this group, or biomarkers that could enrich this group of new-onset diabetes for type 3C diabetes, this would yield an even higher risk group for pancreatic cancer that could be screened using imaging and all of the current methods for identifying pancreatic cancer. Of course, a biomarker that could accurately detect pancreatic cancer early would also enrich this group for pancreatic cancer, and those individuals who test positive for such a biomarker could also be screened. So our own work, based on mass spectrometry, proteomic approaches and validation using mass spectrometry and/or ELISA assays, allowed us to enrich or study proteins that are related to diabetes. And we identified candidates for validation, leading to the identification of 2 proteins, adiponectin and IL-1 array, that distinguish type 3C diabetes from new-onset type 2 diabetes. Our work was undertaken with samples from cancer patients who had already received their diagnosis of cancer. So how do we take such markers that are promising and ask what their performance would be in the months and years leading up to the diagnosis of pancreatic cancer? For this, we need cohort studies of very well-defined individuals in terms of their diabetes status prior to the diagnosis of pancreatic cancer. So this leads me to UKEDI, the UK Early Detection Initiative for Pancreatic Cancer, which is a programme funded by Cancer Research UK, aimed at detecting pancreatic cancer early in individuals with new-onset diabetes. So this programme of work has a number of work packages. We are recruiting individuals who've been diagnosed with diabetes within 6 months, within the last 6 months. Our target is 2,500 individuals. We're following them for 3 years. We will take samples from those individuals. And that will allow us to validate biomarkers that are promising, showing promise for early detection in the NOD group. We are also following these individuals at epidemiology and taking questionnaire data and clinical information from those individuals. The resource that we are building, which is a pre-diagnostic pancreatic cancer resource, will allow us to undertake new biomarker discovery. We're undertaking cost-benefit analysis to provide an evidence base for policymakers. And of course, we are also engaging with a number of stakeholders. And one of our aims here is to raise awareness of the link between new-onset diabetes and pancreatic cancer. So these are the list of sites in the United Kingdom that are approved via ethics as research sites. And you'll see that we have different types of sites. We have pancreatic referral centres, specialist diabetic specialist centres, and primary care community-based centres. These are the sites that are currently open. And I think you'll note that we have good success with diabetic specialist centres. There's great overlap and complementarity between what they do and what we are trying to achieve. A number of the sites that I've mentioned have affiliated patient identification sites, and these are normally primary care sites associated with the hospital trust. So in total, if we count the sites that I've just shown you and the PIC sites, we have a total of 33 sites open at the moment in the UK, and we have plans to further open further sites as we go. So while UK EDI is in process and participants are being recruited and samples being banked, we are also undertaking a number of other approaches to understand new onset diabetes in pancreatic cancer. And Martin Stott has a poster at this meeting. The number is shown here at the bottom of the screen. And one of the things that Martin has been doing is following the diabetes status of 175 patients undergoing pancreas resection for operable pancreatic cancer at Liverpool between 2016 and 2020. And the data are shown here. Over 50% have diabetes, 32% new onset diabetes, and 13% long-standing, and a further 12% have impaired glucose regulation. One item on the poster that I just want to draw your attention to is the fact that of the individuals with new onset diabetes, almost half were unaware of the fact that they had diabetes. It emerged during preoperative screening. So there's a lot of work to do yet in terms of awareness amongst the population and also the clinic in the clinical setting. Martin is also presenting data on insulin use preoperatively and postoperatively, as well as other forms of diabetic medication. We are also continuing our biomarker research. So in collaboration with the Stoller Biomarker Centre in Manchester and the Somologic group in Colorado in the United States, we have two studies that are currently ongoing. So both those centres now have our samples and are analysing them at the moment. We have 210 samples comprising 7 case and control groups. So our case and they're stratified according to diabetes. So we have pancreatic cancer, chronic pancreatitis, groups stratified for diabetes or no diabetes. We also have control groups of individuals with new onset diabetes and individuals with long-standing diabetes, and of course healthy controls where there's no, where they do not have diabetes. And we'll be looking at blood proteins. We estimate to quantify around 7,000 proteins per sample. And the research objectives here are to find circulating proteins whose levels are different in type 3C diabetes compared to type 2, to observe differences in the proteomic profile of pancreatic cancer-related and chronic pancreatitic-related diabetes, and to explore pathways associated with type 3C diabetes. This point is very important because although we are using diabetes and intend to understand how to use diabetes to detect pancreatic cancer, in actual fact, the understanding of the biology underpinning pancreatic cancer-related diabetes is poor. So how does pancreatic cancer cause diabetes? Why do some pancreatic cancer patients develop diabetes and others not? Are pancreatic cancers that develop in the presence of glucose dysregulation different from those that do not? And so to address some of these questions, we are also in parallel undertaking taking tissue samples from pancreatic cancer patients who did or did not have diabetes and undertaking single cell analysis for RNA sequencing. And we will endeavour to combine our single cell RNA-seq data with our SOMA and SWATH circulating protein data to build up a picture of pancreatic cancer-related diabetes. And we hope to identify and validate genes that are differentially regulated in diabetogenic pancreatic cancer versus non-diabetogenic pancreatic cancer. So in summary, I've talked a little bit about presented you with some of the published evidence regarding new onset diabetes and pancreatic cancer. I've directed you to some cohort studies that are ongoing in the US, Hungary, and the UK, and one of those is a randomized controlled trial aimed at understanding new-onset diabetes for early pancreatic cancer detection. I've shown you that the blood levels of adiponectin and IL-1RA distinguish type 3C from new-onset type 2 diabetes. I've talked you through the work packages that are associated to our own cohort study in the United Kingdom called UK Early Detection Initiative, and pointed out that almost half of individuals found to have diabetes or with new onset diabetes undergoing pancreatic cancer did not know about their diabetic status until preoperative assessment. I've also mentioned the fact that we are continuing to undertake extensive analysis for blood and tissue to further dissect pancreatic cancer-related diabetes for early pancreatic cancer detection. So I would like to stop there and thank the UK EDI team, the Trial Steering Committee, and the UK EDI Management Group. Lucy Oldfield and Martin Stott generated the biomarker data and the clinical characteristic data respectively presented here. Finally, I would like to thank our funders, Cancer Research UK and Pancreatic Cancer UK, for funding the work that was presented here, and say thank you once again to the organisers of the meeting for giving me the opportunity to present our work at the CPC meeting. Thank you very much.
Thank you very much for this excellent talk. We have a couple of questions, and Jason typed that he would like to ask something. So please, okay. Jason.
I thought it would be easier to ask directly rather than type. So fantastic talk, Ethne. Really, really interesting. So I was really interested by the fact that you showed that the risk of PDAC was much lower in long-term type 2 diabetics compared to new onset and possibly type 3C diabetes. So does this suggest that it's the loss of insulin secretion that is effectively increasing the chance of PDAC, perhaps even, facilitating PDAC, as opposed to hyperglycemia?
I think that my understanding here, Jason, is that actually pancreatic cancer is causing type 3C diabetes. So it's causing the diabetes, and the fact that, so if you are newly diagnosed with diabetes over the age of 50, there's a 1 in 100% chance that you have a pancreatic cancer that has caused the diabetes. So that is why the risk is much greater in the 1 year post diagnosis with diabetes. The risk of pancreatic cancer is much greater than if you have diabetes for a long time, because if you live past 3-4 years with new onset diabetes, it probably was not caused by pancreatic cancer.
Okay, so any of the patients with the new onset, did any of them have any bouts of acute pancreatitis? Could you tease out that data? Because that would be interesting.
That's interesting. That's not something we've looked at, but that really is interesting. We have no data on that, but it's something we should look at.
Because presumably, if you're talking about type 3C, most type 3C cases are caused by pancreatic injury, presumably due to an acute attack of pancreatitis.
I think 67% of type 3C diabetes is secondary to pancreatitis. Oh. I'm not sure it's acute, it's chronic as well, chronic pancreatitis as well. So most cases, 10% of type 3C diabetes is caused by pancreatic cancer.
Okay, thank you.
Yeah, thank you.
Thank you. I think Federico also has a question, so I think it's just much easier to ask directly and keep the discussion ongoing.
There was also another question before mine by Professor Krivaruchko.
Oh, yeah, right. So there was a question in the chat. How many years can diabetes preceded the manifestation of cancer in the pancreas?
I think the data that Suresh Shari has generated on this subject indicates around 3 years. So on average, the time from, yes, so diabetes in pancreatic cancer patients develops very quickly, as opposed to diabetes in type 2, in regular diabetes, or the more prevalent form of diabetes. So my understanding is that it's much more, it proceeds much more quickly. So 3 years prior to the 3 years prior to pancreatic cancer diagnosis, hyperglycemia begins, and at about 12 months prior to the diagnosis of pancreatic cancer, tips into diabetes. So hyperglycemia tips into diabetes. So it's got a much more steep progression from hyperglycemia to diabetes if it's pancreatic cancer related. I hope that answers the question.
Yes, it does. Thank you very much, Federico.
I wanted to ask a little bit provocatively, but I think you almost more or less already answered it. So at the beginning, you showed that it wouldn't make sense to screen for pancreatic cancer in the general population because it's rare. So of course, it would, even with a fantastic test, you will have a lot of false positives. So I was wondering, if you limit to new onset diabetes, that's still a lot of people. So even with an excellent test, you would still have a lot of probably more or less the same problem, just on a smaller scale. But probably the way to go is really 3C, type 3C diabetes, because as you just said, a large proportion, you say 10% of those have pancreatic cancer. So of course, in that particular group, it makes particular sense.
That's exactly right. Everything you've said is exactly right. So it's why even when you take the new onset diabetic population over 50, you still need to enrich it in order to start screening. And one of the approaches that we've taken is to say, can we separate out the type 3C individuals? Because then you get down to 10% of the type 2 group, 10% of the new onset group, and 10% of those has the disease. So you're in territory where it's reasonable to screen if you can do that.
That the numbers make sense. Okay.
Thank you very much. I think it's time to move to the next presentation because we are almost on time.
All right, so I think it's back to me to introduce our final speaker of the session, who is Peter Hegi from Semmelweis University in Budapest, Hungary. Peter has a very ambitious and comprehensive title, Translational Research in Pancreatitis from Bed to Bench to Bedside. So Peter, please.
So, ladies and gentlemen, first of all, thank you very much for this prestigious invitation to speak at the European Pancreatic Club meeting. Thank you for our Ukrainian friends that in such a difficult situation, they decided to go on with the conference. I must say that all of our heart is with Ukrainian people, and we hope that peace will come really very soon in Ukraine. So today I will speak about a little translational science from basic science towards clinical part. But first we must understand the clinical issues of pancreatitis. One thing is which is threatening that the incidence rate of acute pancreatitis continuously elevating. However, research on pancreatitis continuously decreasing, and the mortality is still unacceptably high in the severe form of pancreatitis. As you can see on this slide, that unfortunately, currently we don't have specific therapy in acute pancreatitis. To get solved this problem and to have therapeutic targets and new therapeutic interventions, you must start at basic science, go through applied science and clinical science. Therefore, we first went for the basic science part, and we wanted to understand that how does acute pancreatitis developing. So our first part of experiments wanted to understand how the disease develops. However, first we must understand the basic physiology of the pancreas. The pancreatic acinar cells secrete a large amount of bioactive molecules, including pancreatic enzymes. However, pancreatic ducts secrete bicarbonate-rich isotonics and alkaline solution to the pancreatic lumen, and these two kind of cell types provide approximately 1.5 liters of fluid daily. If we look at an electron microscopic picture of the two cell types, you can see a huge amount of mitochondria in both cell types. And they, of course, we know that secrete lots of energy, which is essential for physiological secretory processes. So let's look at how pancreatitis is developing. One of the most common etiological factors is the biliary part. And my first PhD student, Victoria Wenglovec, was the one who performed this kind of experiment. And actually, she mimicked the situation. She injected bile acid into the pancreatic duct tree and looked at its result. And we could show that bile acids in a low concentration stimulate, however, in a high concentration, totally inhibit the pancreatic ductal bicarbonate secretion, which actually, due with a calcium signal pathway, low doses of bile acids inducing both from the basolateral and luminal side an oscillatory physiologic calcium signal. However, when bile acid arrives to the cells in a high concentration, it causes really toxic calcium signal. So all of our experiments showed that not only the bicarbonate transport is stopped, but if you look at the mitochondria, they are irreversibly damaged, which actually resulting decreased ATP secretion. So we wanted to see whether the decreased bicarbonate secretion is with intraductal acidosis as well. So we perform a pH measurement inside the pancreatic ducts, and we had 6 acute pancreatitic people, and actually the control groups were those patients who had to undergo ERCP. And we could see that during acute pancreatitis in the pancreatic duct lumen, there is a clear intraluminal acidosis. And if you look at also the time dependency of the process, as long as the blockage of the outflow of pancreatic juice occurs, the rate of pH decrease is higher. So altogether, we must say that in bile-induced acute pancreatitis, there is a clear decrease of ATP secretion, toxic calcium signal, mitochondrial injury, and decreased bicarbonate secretion, resulting in acidosis and blocked trypsinogen secretion by the acinar cells. Let's look at the other kind of etiological factor, namely the alcohol-induced or the fatty acid-induced pancreatitis. Actually, what we could see that alcohol in a very small concentration stimulate, however, larger concentration inhibit the pancreatic ductal bicarbonate secretion, and especially if you see the CFTR chloride channel, this is very clear that both the effect of alcohol and the fatty acids are via CFTR inhibition. And this is also like in a bile-induced way, coming with ATP decrease, mitochondrial membrane potential decrease, and also coming with inhibited luminal pancreatic secretion. If you can see a closed model of measuring the volume secretion, you can see in a green one, and the black one is actually with the controls one. And if you give alcohol or fatty acid, this is tremendously decrease the fluid secretion. This is also confirmed by MRI as well. So finally, if we look at this picture, it's very similar to the bile acids one, that alcohol and fatty acids strongly decrease mitochondrial function, decrease ATP secretion, toxic calcium signal, blocking CFTR chloride channel, and causing intraluminal acidosis. So this kind of energy suggests us that what can happen if we restore the mitochondrial function by giving ATP back to the cells. So the next kind of experiments, both our work group and also the very famous Liverpool work group, try to replace ATP back to the cells. And we could see both the acinar cells And both the doctor says if you replace the intracellular ATP, the patch clamp technique, you can at least partially restore the function of the cells, which actually suggests the therapeutic benefits of giving energy back to the cells. So the next question was that do we have enough evidence in clinical science that early energy has beneficial effect? Usually we start the clinical science part with the meta-analysis, and Katalin Marta, who performed the meta-analysis, collected huge amount of papers both in severe pancreatitis and mild pancreatitis, and the results very clearly suggested that both in severe pancreatitis and mild pancreatitis, early restoration of energy level by enteral feeding could be beneficial. So the next part always prospectively and retrospectively looking at our patients cohorts, and we could see in our cohort that if we use nasogastric feeding, it can actually decrease the severe and moderately severe range of pancreatitis. Whereas in the Hungarian cohort, in those patients where the patients get no energy in the early phase, it's still the length of hospitalization was 2 days longer than in the other part where the patients receive lower energy. Actually, in these 200 patients, this could generate 415 days less hospital stay for the patients. And the other striking data was here that also, besides that, the length of hospitalization, the mortality was also decreased by 2/3 using a very early energy feeding, and also the necessity of antibioticum usage were less. So actually, these data suggest the beneficial effect of early enteral feeding. However, always the final confirmation in clinical science is actually randomized clinical study. So the Dutch pancreatitis study group performed a large study in which they wanted to see the effect of early versus on-demand nasoenteric tube feeding in acute pancreatitis. However, if you look at both groups, neither of the groups received in the very early stage high amount of energy. Therefore, currently no sufficient amount of energy was administered here. So this study protocol was not suitable to make decisions on high calorie administration in the early phase. Therefore, we established the Guyash study, general utilization of energy back to the patients, and we used a very similar protocol but the Dutch pancreatitis study group used. However, we started in one group with a high energy, and actually in the other group, we continued with the step-up approach, building up the energy intake for the patients within 3 days. In this project, we are at the halfway. Approximately 450 patients are already involved. So the first interim analysis will come within 2 months, and we will see whether how to continue actually this study. So this is a very important aspect, that how to find therapeutical approach as basic science, applied science, and clinical science important. So we hope that also finding another targets, we may have a specific treatment in pancreatitis actually in the near future. So finally, I would like to thank you again for this very prestigious invitation, and hope that peace will come very soon in Ukraine, and we can meet also personally in the near future. Thank you very much for your attention.
And thank you, Peter, for this interesting, comprehensive presentation. So I see Jason Bruce has a question for you. So Jason, please.
Okay. Again, a very interesting talk. Thank you for that, Peter. you suggest that the enteral feeding or early enteral feeding has a beneficial effect, and that might be because it's providing the pancreas with energy. However, if I remember correctly, and correct me if I'm wrong, enteral feeding is more beneficial than parenteral feeding. So could this be because applying the nutrients directly to the gut is having a much better beneficial effect, either because it's altering the gut microbiome, or it might be directly activating enteroendocrine cells that are secreting, for example, incretins, GLP-1, which are more effective at activating insulin secretion than glucose alone. Could that be the explanation?
So thank you very much, Jason. This is the forthcoming next application form with all aims, you put it in. A brilliant question, of course. I mean, we have to state that all pancreatic cells, including beta cells as well, most probably, they are lack of ATP, lack of energy. And of course, they need energy. From this aspect, you are completely right that alone parenteral feeding would have the same benefits. However, we all know that enteral feeding has other tremendous benefits, especially the microbiome inside the gut. On the other hand, one of the biggest causes of mortality in acute pancreatitis is pancreatic infection, which is usually going through the gut system. So locally giving energy to the cells, because during the absorption part, you directly can drive actually also energy to the gut cells and the small muscles and everything. You keep the epithelial cells barrier with the tight junction correctly. So I think this study is really a comprehensive one. However, because of this comment, the control group also get enteral volume and the same way. The only difference was and with the same ion concentration. The only difference between the two arms were actually the amount of calories and of course the protein, fat, and carbohydrate levels. So we try to mimic the same situation for the control group, but yet we don't know whether the elevation of incretin or the others, what we call, actually we keep all the blood samples, serum cells, plasma, in a time-dependent way from 1, 3, 7, 14 days during the interventions. So most probably your question will be answered when the study primary endpoints, which is the mortality and severity, are closed. There will be lots of additional studies will come involved from this trial.
Okay,
thank you.
Great, thank you.
Thank you. So I don't see other questions from the audience. Any other questions from the panel? Peter? Okay, if not, we are...
I mean, just I have one comment if we have
please.
time concerning,
Sure.
because the title was about this translational medicine. I think in the 21st century, it's very, very important to form translational research work groups, because you must understand the basic part of the interventions you would like to bring in, and you must understand from the clinical sides and collect clinical samples. So exactly what you are doing with Pandora and all the other parts, I think it's crucially important, and we should even highlight the importance of translational research groups worldwide, because I think if I look at pancreatic groups, there are plenty of places where only one of them existed, and which is, I think, not beneficial nowadays. If we look at also the Liverpool's big success with Alexei Etny and all the other parts, this is also was the biggest secret, is to get the work groups through together. So maybe my presentation is not only bringing the attention for acute pancreatitis, but also the multidisciplinary research teams.
I think we can only agree with all that.
It is a good point.
Okay, and on this, I think we should conclude this session. So thank you very much to all the speakers for very good presentations and interesting discussion. So I think now there is a pause for 10 minutes, and then at 5 Central European time, there is the next session on pancreatic endoscopy. So thank you very much. Bye-bye.
Thank you. Bye.
Bye.